like that. Well, you never know.
No collections.
Collect raisins.
Oh, razors. I collect razors.
But you actually use those.
No, I like the old fashioned
Like the shaving kits with the little.
Yeah.
With the brushes.
brushes.
and-
The brushes.
Those are pretty cool.
Some of them are really old.
What do I collect?
And they look kind of pretty.
I collect rocks, seashells, and plants.
Plants.
I don't have too many plants, actually. There are only so many that could fit in the house.
That was one of the first things Mel noticed when she came here was you didn't have any plants. No.
No.
Well, the plants in my office die because they have to sit on that windowsill and the temperature fluctuates too much. But I have plants at home. I have as many.
Office plants.
Yeah.
I'm not going to do anything until this remodel's done. No sense in bringing a bunch of stuff in and then taking it all back out.
So
So
you'd have to take my just one out.
But like a plant, like how
that could be one extra trip downstairs.
I wonder what they're gonna do with my enormous jade plant, because that thing needs to be kept, the one that's in 302. That's what
Oh,
my
yeah.
plant collection is, but most of them were victims of the pandemic.
Oh, yeah. Yeah, that one survived.
But that one survived,
It's got to survive the remodeling.
so it's got to survive the remodel. And at home, I keep as many plants as my husband will let me. He likes them, but he doesn't want any more. I don't blame him.
Can never have too many plants.
Nope.
Yeah, some plants probably would help your office and stuff.
Yeah.
They don't do real well in our offices, as you can tell.
Yeah.
And you guys have stuff to keep them on away from the windows, and that'll be good.
There's a hot rubs in the windows and things.
Yeah.
No, I'm not going to do plants. I'll fill mine with hides, skulls.
Well, it's kind of cool too.
Yeah, that works.
Just, you know,
Yeah,
stuff.
shells.
Yeah.
Stuff.
Posters.
We have giant stuff. It was hard.
Yeah, like right on your desk.
Yeah.
So you have to like look over it. Like a real alpha symbol.
My old advisor at Tech had a stuffed, I forget what it was. It was a squirrel or something. And it was the ugliest, most amateur taxidermy job I think I'd ever seen in my life.
Maybe that was the point.
He didn't buy it. It was like given to him by his mentor, and then it was like this little log. It was about that big that sat, and there was a squirrel on it, like holding a nut, but it's like like this. It was just the
Like a real
weirdest thing.
natural pose.
Yeah,
Like that.
yeah. And its mouth was all messed up, and fur was gone.
Go easy on it. It's pretty sweet.
It's pretty sweet.
You can get a cow to go with your tractor.
You could get like a little barnyard set.
Could have my own farm set.
Just have it on the desk. People to come in and play with.
Well, I brought that in here week 2 of block 1, and it's lived in here ever since. So nobody's moved it.
Tractor.
Yeah, I don't understand.
Oh, that's nice.
That Kyle has a tractor.
Tractor. It's just the bits are kind of. I don't know if that's age or somebody's been picking at it. It's brand new.
So unlikely to be aged.
Yep.
You don't know.
A lot
I
of
mean,
students have probably played with it.
Yeah.
This is true.
age is relative.
I know we brought some people joy with that.
Yeah, it just lives there.
I sent in the request.
Thank you.
Well,
Like that I don't like need a pointer or anything.
No.
Just be like point.
This isn't that kind of place.
Where you're like,
oh,
you
university.
see that up there?
Nice not to, either be preparing.
There's
surgery
a,
or recovering from
A
surgery.
COVID stick back
Break
there
is
you can
good.
use
Break
if
from
you want.
surgery is good.
Oh, yeah. Oh, we used to have
Prefer
these
never to have
sticks,
any
so
ever again.
very few people had
Hopefully this is
And
the
in
end.
306, there's
So,
a
both
tip
of
of a
these
fly
were
fishing
injuries.
rod that I walk around with. So
That's just it's like a
Yeah,
little.
yeah, take it. Take it
Slow
easy.
down.
Yeah, exactly. A little less extreme sports.
it's great though because it has a rubber tip at the base so
Although
instead of
one of
dropping
them
it
was
on the
made in
floor
my dad's.
and letting it bounce,
The other
or else
one
I
was
stand there.
Skiing?
What's that?
What was it, skiing?
Yeah. Yep. Tell them
I
I'm
fiddle
good.
when I do things.
Not gonna do that anymore. I'll still cross country ski. That's it.
Erica,
I'm not going better.
Erica, it's my partner
Nice to
Mel.
meet you, Erica. Nice to meet you. Thanks for sharing. Oh,
Hi,
so-
Erica.
thank
Hi,
you.
how's it going?
Good. Just coming up the stairs.
Nice to see you.
You too. Snacks.
Well, where
This
is it?
is our, it's actually going away because we never
Local.
do anything where we can fill the room. So in the remodel, this is going to be broken down into sub-molecular rooms. So this will either be 2 classrooms or labs. I'm not sure which way. I think they're doing classrooms here.
I don't
Lab,
know.
and chemistry will be there.
Maybe?
So they'll have a
Okay.
classroom with us on lab space.
All right.
I'm not supposed to tell you how they're made, but Kyle.
He melted a bunch of chocolate and added caramel.
So,
That's what I understand.
Yeah, I described it.
they're-
You mean from dried powder?
For sitting, especially for-
Yeah, basically.
Oh, was it dried powder?
It's sad. Moving. Great.
No.
Oh,
I have to find somewhere that's
okay. So it was reconstituted, you're right. You probably added a little milk or butter or
I
something.
wasn't being facetious. Trying to be active.
Yeah, this whole place is gonna change. I think we're gonna try and do more windows at the back.
Okay.
That'd be nice.
Just like the oil.
Well, it was a chocolate bar
For those
like this. This brand. It was shipped from New Zealand. Kyle didn't.
It was basically like
It's repurposed
have
chocolate.
to throw it out sort of chocolate, but.
The old chocolate?
That's the old
Yeah.
chocolate.
Oh, we
that's
rest-
better.
Salvaged it.
If you melt it down and add some cream and then cover it in a boatload of caramel, that's the secret.
It was do that
so
or
bad.
throw away like 5 kg of chocolate.
Did you see it before?
I gave her a block.
Oh, it was just like powdery. It was so, it's not supposed to be like that at all. So the dark chocolate was actually okay, but some of the milky ones were just like dust. Pardon? Yeah, I think it was the more milkier ones that like really suffered, but the darker ones were like pretty much okay. So I just, well, I think he was saving the dark ones for himself. He didn't know they were crap until I think he just gave them out and then he went home and tried to eat one and he was like, oh no, this is very bad. But he'd like filled, he had all these boxes and one of them was just had like 30 bars in it or something. Like it was just like, I'm gonna take all this chocolate. He took some in his suitcase and then he sent some so he'd get it like 6 months later, which is a really nice idea. And then when he opened it, it was just like they're all ruined. I was just very sad for him. So I brought him a lot of bars this time. I was like, don't throw the old ones away. I'm sure we could salvage some of them. I was sure he hadn't opened every block, but we probably threw away 2 blocks worth, but all the rest were they're fine. When you just put a lot of work into them. He's got. If you want some of the fresh one, there's a new one there too, which it's got honeycomb in it. Actually, I don't know what you call it here. For some reason, he's called it.
You guys have got to. What do you think?
Kind of low texture it was originally.
It's pretty good.
What? How is this a creamer? Such a strange country. No.
I didn't know that was
What else
possible.
did I eat that only one that was cinnamon? Okay,
No, but
I'll go with cinnamon stick.
I could not know this.
Slackers in the back.
Do you know Ezra Preston?
I know it has a regular shooter down
Dark
there.
hair.
Yeah, dark hair.
Hi.
Yeah.
There.
I wasn't paying attention. I was looking back at the clock and suddenly you were like,
Oh, you were. That's pretty much like in between a mom and their cubs. You're
General
between me
science
and the dog.
education.
One of my favorite.
Yeah.
it's just lemonade.
I don't know. Give it a try.
No, I don't want to.
Yeah.
Oh, okay. Then you just have to remain in the street.
So I would have to sit really far away from the speaker, or the speaker would have to talk.
Or they just tiny little things.
Yeah, they looked like desert succulents, just covered. There was one that was super, had all the great filamented structures coming out of like this.
That's what it would wrap around. Just kind of thing to hang on.
But that was like the main structure. It didn't have anything coming out of the chest, and the others didn't have anything for moving on the branches. It just looked like little. The one of them had like the leaves kind of came out almost like I guess the best way is try to do like succulent leaves, but it looked they kind of came out almost like pineapple.
Oh, yeah, pineapples are bromeliads. That's the only bromeliad that is actually, canonically important. It's a pineapple. So that's why it looks like a pineapple. Same family plant.
Have them do a design pretty purely.
Yeah. So they look like little tiny pineapples.
They didn't have like a pineapple fruit, they just looked like the pineapple leaves.
Yes, that's what I meant, the leaves, yeah.
They have to roll.
People back home who some of them used to work with more of the trusted vendors. So they're that kind of family friends, but they're originally from Hawaii. They talked about how there's this pineapple there because some of them were pineapples causing ulcers and things like that. So there's this pineapple called white pineapple that they say grows seasonally in Hawaii, but
It's a fairly straightforward.
it's very short growing season. It doesn't really last long enough for them to.
I've never had it before.
But apparently it's my father said it sounds like it's less strong, more of a subtle sweetness,
And I got
less
smoothies.
acidic.
Well, when we do our angiosperms unit, I just try to bring in a whole bunch of flowers and plants.
Or is it even
It's probably.
Yeah, that's what I told him. About like, it's like the two of them didn't find it out. Timing. Rob was just gonna go on after the.
No, I was just if it's that year, not the previous year.
So,
I got it all, so I'm aware of the timeline.
And then we do the flushing.
sorry.
I didn't do the medication, I just did Tylenol and ibuprofen. And honestly, like all this ibuprofen and Tylenol.
Probably working until the grant we're looking at.
I'll go like the high, and I finished the
I gotta build research here,
No,
because I can't keep going.
no, this is a classic New Zealand. Yes. Not sure what it is, but they've got terms for everything, so it belongs in there somewhere.
Are you good?
How's that going?
Our crow. Learn on our crow.
Oh, you did.
I took GIS in 2010. I mean, I'm learning it. I mean, it's all the same. It's like just repackaged.
Oh, okay.
But not too bad, yeah.
Tough crowd
One
here.
of the IT guys was telling us how bad the GIS program is.
Oh, because he was the GIS person. For Dylan. Yeah. Well, we have Pro, which is what the industry
He
stands.
said the license is expired.
I had to do all the updates. He's like, I can run it from my like city position, but not from the university position.
You do be careful if you touch it.
But, I got it up and working.
Arrow keys.
Not all my phones.
Just making sure you. If you want, we can hook up the other two screens and fix around. It is, but Bradley felt like he needed it.
That's right.
What?
Just talking about hooking up the other two screens.
You want more displays? We can
This
make
is
triple.
pretty big.
We make that happen.
I feel like.
Can you guys see it and reading it in the back?
Look at the IMAX.
Can you see what's on the second line?
Looks like anatomy.
No, second from the bottom.
Second from the bottom.
Yeah.
That's an outside, isn't it?
No, it's not.
It's like an eye test.
But if I don't go up these stairs, I feel like about a
I think all my writing is bigger than that.
Okay. Sorry, Dr. Lord.
I don't know why. My freshman year, me and my friends group just before had nothing to do, we complained about the same. So, but a lot of my friends, they really.
Just read out small writing is right.
Yeah. We came in.
We can roll this forward.
That might be good. I'm gonna let someone else do that.
All of the stairs.
Still gives me enough room to
How far forward do you want?
I don't know exactly. So I'm not gonna fold when I. Actually, I still have.
Is that gonna mess up your camera angle?
I can't remember if it was a centimeter shorter or inch shorter. These were shorter than the ones in that. That's it, that's the only difference. Yeah. Green hole stairs. One that you did that. Why are you doing those stairs? We were poor. We were actually like we had nothing to do. I've never been like, I'm bored of measuring stairs. We had nothing to do. I'm bored. I'm gonna go. Measure the stairs, and I think my best friend who's now trying to get NBSU.
Wind down for their Wednesday.
I think she has enough, but we wrote down, because we were all like in the same gen ed class. We were all in the same gen ed class.
And this is what Federation used to have, another row. That right there. So
But for microbiology, Dr. Morrow, find something,
well, yeah, our classes are capped. And I've never seen this room filled like she's been told that.
So what I see there is reverse organization.
You never
It's super.
filled this class up. So that's why it's going well. I think we're going to put a similar room to this on the second floor because that's math. That's the reason. Yeah.
Seems like that's something.
Try and do it very quick.
Well, I guess.
Neil.
Oh,
What's up?
oh, not much. How are you?
It's my partner Mel. Mel,
Hey.
Neil. Nice to meet you.
Looking forward to it. Hey, you're back. How you doing? All right. Here. Yeah, happy to meet you guys. How was break?
Busy.
Yeah.
Busy. What'd you guys do?
Apparently I went back to East Coast, saw my folks for a bit. She went on to Cincinnati, and I was like, I want to go to Cincinnati, so I just stayed home with my folks. And, it was lovely. I did actually very little.
I reckon that is a good choice.
Yeah.
Not going to Cincinnati.
Yeah.
Okay. Or I can do it
Cincinnati.
tomorrow.
Riley saying you were maybe thinking about going to the brewery after this? Or?
Yeah. Yeah. Hopefully the Mexican place is open. So.
We were possible now.
Sweet.
Yeah. We were going to do that at one point and then go to the brewery. We went and it was closed, but of course I was like, kind of right in the middle of the holiday type deal. So yeah.
They're just, I don't know. You look at the door, they're supposed to be open like 24/7.
Yeah, sweet.
Not
I'll
actually
swing
what
by
they
and.
Okay.
Yeah.
Sometime
No,
in the
we'll walk
afternoon,
over after.
maybe 2.
We'll wind
Or even
down
I can come.
after a stressful talk.
Okay. Cool. Sweet.
For you.
Thank you.
Why?
Okay.
Hopefully it's not stressful for the.
Don't worry, he's willing to drink a beer on that. Real team player.
So where's the?
Looking forward to it.
I'm gonna grab a cookie though. Grab some of the chocolate and the Tupperware too.
Fudgy stuff.
Ooh,
Fudgy stuff.
This is homemade?
What'd you say?
Is it homemade?
Yeah.
All right.
Things are getting close.
Sweet, so it can go easy.
I think that reacting, that's how effectively they're reacting is the question.
Yeah, they're wrong. Would you rather be in the library?
PTSD responses to that. No, they're all one set.
It takes me so long to like.
True.
That's what I'm saying.
Won't interrupt.
How long do you think?
Probably.
And I can go here.
Right. You. I'm sure there'll be questions.
Yeah, great.
You know how to.
Hello. No, I didn't give an ultimatum. I just told them.
I figured he was like, come or I'll fail you.
No, he didn't do any of that. He was just like, I'm not gonna spoil anything for you, but I have some kind of talk about this.
So it's been like set up. So now I have to deliver.
Wow. Most of the students.
Many people go to
And then he got frostbite. He got frostbite. Yeah. Yeah, so he kind of, yeah.
like people just came in or something.
Yeah, for a little bit.
Well, I told these guys that if they had questions to ask you, we'd go over it again tomorrow. Some issues.
Oh, he's where right now?
Yeah, come in at like 10:30 or something. Stroll in about 10:30.
Good. That's what I need to hear.
Right. Make it 100. You're gonna be 24.
Oh, you're gonna do that. I told him once he started working out, so you're gonna start paying my bills, right?
Get this thing going?
Yeah, sure.
Are they?
All right, let's get going. 4 o'clock. Thanks everyone for showing up. This is one of the best turnouts we've had so far. I'd like to introduce Dr. Melanie Laird. Melanie did her PhD at the University of Sydney, working on mammal and, more important, marsupial reproduction. Now she's at the University of Otago, where she's working on marsupial development, and she's here to talk to us about the other mammals. So, good.
Thanks, Kyle. And thanks everyone for coming. So I'd like to spend my time with you today, which I'm very grateful to have this opportunity to speak to you. I'd like to tell you about what is my favorite group of mammals, called the Marsupials. See how we can work this not awkwardly. So I firstly like to acknowledge all the different institutes and particularly funding bodies that have supported the work that I'll present to you today, including those from Australia and New Zealand, where I'm based now. I don't have a pointer, so I'm going to awkwardly just sort of like gesticulate. So, as Kyle sort of introduced, I'm a research fellow at the University of Otago, and I wear quite a few hats. Can you guys hear me at the back okay? It's quite a deep room. So I'm primarily an evolutionary biologist and a comparative biologist. So I'm really interested in sort of comparing and contrasting different groups of particularly vertebrates to answer big evolutionary questions. And the questions that I've mostly focused on are those related to reproduction and development. I feel like maybe this is a bit...
Do you want to move?
I just can't see this. Yeah, let's move it back to where it was. So I did my undergrad and my PhD at the University... Oh, that's a good way to do it. Thank you. At the University of Sydney, which I really like. You can't see this at the back, but Google describes it as an elegant university, which is quite true. I think it's quite nice. And then in 2018, I jumped just across the ditch to the University of Otago in southern New Zealand, which is also quite elegant, I think. So what I'd like to do is introduce you to this group of mammals, the marsupials, and I'll take you through a few different ways of, I guess, defining what a marsupial is. And the first of these is that they're a group of pretty iconic mammals, the vast majority of which occur in Australia. So you might be familiar with the koala, the wombat, the kangaroo, the Tasmanian devil, probably not the brush-tailed possum, but I'll come back to that one a bit later in the talk. Here are some other ones that, well, this is a tree kangaroo, that's a thing. And then we've also got Didelphis virginiana, which is at the North American opossum, the Virginia opossum. I think you guys just call it a possum. And so this one is quite interesting because it's one that you might be familiar with, and it's the only one that occurs north of Mexico. So there's more than 300 marsupial species, which is actually, they're quite a diverse group, and over 70% occur in Australia and sort of New Guinea. But a few occur in South America. This is the only one that occurs in the Northern Hemisphere. So we used to have this amazing species called the Tasmanian tiger or the thylacine, but that went extinct in about the 50s. And then the quokka, which is widely reported as the happiest mammal, and it's our favorite for celebrity selfies. So I really like that one. So that's one way of, I guess, defining a marsupial is an Australian mammal, a cuddly looking, cool mammal. But marsupials are also really interesting to me because they're a separate evolutionary clade of mammals. So what I've got over here is all of mammalia. So you probably are familiar with the eutherian or the placental mammals. So that's the group that we belong to, as well as most other things you can think of: cats, dogs, whales, everything else. Eutherian mammals are sister group to marsupials. And they also have this other kind of strange cousin called the monotremes. And monotremes are echidnas and platypus, pretty much, that's it. And they are a distinct group because they still lay eggs, which is what the mammalian, way down here, the mammalian ancestor used to do. So they still lay eggs now. And so what links eutherian, I'm going to call them eutherian mammals from now on, placental eutherian mammals and marsupials, is that they share this trait of live birth. So instead of laying an egg, we give birth to an offspring. So that's kind of a mammalian novelty. Other groups do this too, but not quite in the same way that mammals do. So even though these two groups are sister groups, they've been separate for a really long time, for over 148 million years. And that means that over that time, they've accumulated some pretty different traits to solve the shared problems that they have of being mammals. And the biggest difference that they have is in their reproduction. So what I've got along the bottom here is, I want to say, a typical eutherian pregnancy. It's a human pregnancy. It's not really that typical, but compared to that of a marsupial. So the pink shading represents time spent in utero. So with the human pregnancy, we've got fertilization here all the way up to birth, and then sort of a neonate or a newborn at the end. If we compare that to a marsupial pregnancy, what we see is that they have an incredibly short time spent in the uterus. They have a really short pregnancy. So the time between fertilization and birth is usually 2 to 3 weeks. It's incredibly short. So you can probably imagine that if you're only gestating for that long, you're pretty underdeveloped at birth. So marsupials are born at the human equivalent of about 2 months into a pregnancy, which is incredibly altricial or underdeveloped. But they spend this really long period of time in an external pouch, and that serves as kind of the second half of pregnancy for them. So by the time that they leave the pouch, they're kind of at a developmental equivalent as something else would be born. So I just want to take you through that short pregnancy and that long lactation in a little bit more detail. So marsupials hold the crown for the shortest pregnancy of any mammal, which is this guy here, the fat-tailed dunnart. It's one of my favorites. So their pregnancy is actually only 13.5 days long, which is amazing. And the other remarkable thing about marsupial pregnancies is that they're always shorter than the estrus cycle. So that's the interval between ovulation. And so in a human, we call it a menstrual cycle. So that would be like a human having a pregnancy that's shorter than 28 days, which is pretty incredible.
So this is just to give you an idea of how underdeveloped these are. So this is a near-term embryo. So it's just before birth, surrounded by its placental membranes. And this is probably a day or half a day later it's born. That's a half centimeter. I should have put it in American units. I don't know. It's really little. And so they're so underdeveloped that most of their organ systems haven't even begun development. So for example, they have such rudimentary lungs that they actually breathe through their skin for a good amount of time after they're born. And so after they're born, after that really short pregnancy, their first job in life is actually a really important one. They have this incredibly arduous task of climbing from the birth canal, from the urogenital sinus up into the pouch. And so that's what this little tiny jelly bean is doing here. Here is the pouch. And so they're helped by mum. So this is a rock wallaby, and she looks a bit weird because she's sitting like this. So she's trying to make that distance a bit shorter for her newborn. And she'll also lick the fur in the right direction to kind of give him or her a bit of a path to follow. And amazingly, I've just told you that these things are really underdeveloped, but what they do have really developed is forearms. So their arms, if you can see them here, are pretty stocky, and they've even got claws, but their back legs, which are kind of hidden by that image, are really just like little paddle-shaped things. So they don't use them at all. And that's because this first task requires climbing motion, so they really need strong, developed arms. And they actually, if you look at near-term marsupial embryos, in ultrasound, you can see that they actually practice this climbing motion. They're doing this in the uterus because that's their first task, and if they fail at it, they will die. And so once they finally reach the pouch, they can suckle. So that's a newborn that would have just made it. And if you're a wallaby, for example, you'll stay there for a really long time. So wallaby has about a 3-week pregnancy, but a 10-month lactation. So reproduction is really skewed to the lactation side rather than the pregnancy side. So this guy, he's still in the pouch. He's probably about ready to be kicked out. He's pretty big. He's all arms and legs tucked in there, but he's almost about ready to be weaned. But there's a huge amount of development that occurs between this tiny little jelly bean and this sort of wallaby at foot. So what I've just kind of shown you, I'm going to summarize, is that marsupials and eutherian mammals both have the same shared challenges of mammals, and they both show this general mammalian pattern of live birth plus lactation, but the way that they do it is sort of a bit different in both. So if you're a eutherian mammal, typically you have a relatively long pregnancy, so a relatively long incubation in utero, and although human lactation is very long, it's relatively short compared to the length of pregnancy. And newborns, perhaps not humans, but for at least some eutherian mammals, can be incredibly well developed, like ungulates can be. Whereas on the marsupial side, they've opted, I guess, for a much shorter pregnancy followed by a long lactation period. And so the consequence of that is really underdeveloped young. So often marsupials are described as sort of primitive or lesser, or doing things in a way that isn't as good as eutherian mammals. I think that's a hangover from the fact that we ourselves are eutherian mammals. But these are just separate but equally effective ways of achieving the same reproductive outcomes. So I just want to show you a few quirks of marsupial reproduction. So a little bit of anatomy. Oh, that's not at all the picture that was supposed to come up. So females have 2 completely separate uteri, which is quite different from us and most other mammals. Even something like a rat has 2 uterine horns, but they're still connected. So in a marsupial, they're completely separate, and each is associated with its own ovary. They also have up to 3 vaginae at any one point. 1 for each. I'm going to just do this with my hands because the picture didn't work. 1 for each uterus, so 2 lateral ones, and then a 3rd transient one will form just before birth, and that's the birth canal. And they all lead down into a single reproductive opening, which is also the same opening for all excretory wastes and urinary wastes. So they just have 1 opening like a bird does, which is pretty different from us. We have 3 if you're a female. So we'll just skip through this because that's a funny picture. So on the male side, males are a little bit different because they've got an incredibly large prostate.
And that's because seminal vesicles, which in us and many other mammals produce the bulk of seminal fluid or semen, didn't evolve in marsupials. So the prostate does the whole job. So it's relatively large. It's this carrot-shaped organ in red there. So that's the bladder at the top, extending down to the penis at the bottom. So in a marsupial, the prostate is responsible for all seminal fluid production, which is quite an important job. And some even have a crazy bifurcated forked penis, which is thought to be able to inseminate both of those uteri at the same time. Can't make this stuff up. Marsupials generally have more active or speedy sperm, and they have relatively long sperm for their body size. So this, again, a picture didn't, it was just literally a picture of sperm. We should have gone through this before, tech person. So this is a honey possum. It's incredibly small, and it has sperm that are 360 microns long. It holds the crown for the longest mammalian sperm, and it's one of the smallest mammals. So its sperm are, 6 to 7 times longer than that of a human, and it's orders of magnitude smaller than a human. So that's a pretty amazing title for something like that to hold. We don't know why they need to be so big, but mammals are just weird. Oh, some pictures that work. Great. So if you're an American marsupial, you also have this really weird phenomenon of sperm pairing. So only one sperm fertilizes the egg, just like in normal mammals. But these sperm have cleverly decided to pair up in order to give themselves a greater chance of reaching the egg. So sperm basically cooperate. So I'll come around this side. So this is just a diagram of 2 acrosomes, so 2 sperm heads that have fused together. And they do this in the epididymis, and they stay together until they reach the oviduct. So this is a micrograph here showing those 2 acrosomes fused. So what you've now got is 2 flagella working together. And they will stay there until they reach the oviduct, and then they will separate and say good luck to you, and off they go. So why would you do this? You're sort of collaborating with the enemy, I suppose. But I guess not surprisingly, if you've got 2 flagella working together, you're much faster than you are on your own. If you're unlucky enough not to be able to pair up, you have absolutely no chance of fertilizing the egg. And it turns out that this is also a really efficient use of sperm. So these marsupials are able to get 50 times more sperm in the right place, so where the egg is, for producing 10 times less. So that's a pretty good return on investment. And some marsupials have a pretty extreme breeding pattern. So a lot of marsupials breed seasonally, like a lot of mammals you'd be familiar with. But there's a few species, one of which is called Antechinus. There's also a quoll that does this, that have this incredibly extreme breeding strategy in which in their whole lifetime they just produce 1 cohort of sperm. And that means that they just have 1 chance of reproducing in their life. And after that, they completely die. So that's why they only have 1 chance, because they're dead after that. And so that means that all the males in the population are gone until that next litter is born. And so this is a strategy called semoparity, which you might be familiar with salmon when they return to where they were first spawned. They breed and then they immediately die. So it's a similar strategy, but it's pretty rare in mammals. And so, like, if you've only got 1 chance to breed, there's going to be this extreme competition for females. If you're not successful here, you won't ever be successful. So there's basically this mating frenzy. And in order for that to work, these males have to tolerate incredibly high levels of testosterone in order to enable that kind of aggressive competitive behavior. So on this graph here, just focus on this, I guess, line of squares that's showing plasma testosterone across the year. So the pink is their breeding season, and around where this red box is when all the males are dead. So you can see that plasma testosterone is pretty negligible for the first few months, but then just leading up to breeding season, they have this incredibly high spike in testosterone. And that's basically what kills them. They die of acute chronic stress. So these are 50-gram mammals, so they're really little, and they're tolerating up to 10 times more testosterone than an adult human male. So that's enough for them to die, basically. But this is a strategy that works. So marsupials are also really fascinating models for biological research.
So you might expect them to be good models for things like reproduction and development and evolution, hopefully, from what I just told you. But there are also a whole suite of human diseases that marsupials are actually better models for than some of your typical model organisms. So these include things like reproductive birth defects like hyperspadia, where genitalia don't form properly during pregnancy. You can actually induce these in marsupials. They're also really great models for things like aging and Alzheimer's, because the way that the brain changes as they age is more similar to our brain than other things like pigs or mice or rats that we would normally study. And Monodelphis domestica, which is the grey short-tailed opossum, it's a South American species, is one of the stars of the marsupial research world because it's quite an easy animal to work with in captivity, but it also has all these weird biological quirks that make it really useful for different studies. So one of these is studies of melanoma. It's the only mammal that we know of that can get melanoma from UV exposure alone like we can. No other mammals can, apparently. And you can also completely sever the spinal cord of 7 and even up to 28-day-old young in the pouch, and they will regain pretty much complete mobility and limb function again. So it's really rare in a vertebrate, let alone in a mammal. There's many, many other examples of marsupials in health-related research. So now that I've introduced you to marsupials and why they're good research models, for the rest of the time, I just want to take you through 2 different projects where I've used marsupials in my research. And the first of these was a study that I did for my PhD on marsupial pregnancy and formation of a placenta. This was at the University of Sydney. Excuse me. So the placenta is an organ that forms only during a pregnancy, and its role is to support the needs of the embryo during the pregnancy. So this is, I've shown you this picture already, but this is a marsupial that's about to be born, and it's surrounded by its placental membranes. So you can see that they're quite vascularized. So that's providing nutrition. It's also providing oxygen, and it's helping to remove waste. And so as this new organ forms, it consists of both embryonic and maternal tissues. So there needs to be this really intimate association between the uterus and the developing embryo for it to form. So it forms at implantation, which is when the uterus adheres to, the embryo adheres to the uterus. And the types of placenta that form are incredibly variable across marsupials, and that's because the ways that an embryo can implant are also really highly variable. So I've just got 2 examples here that are kind of the 2 ends of the spectrum. This first example here is kind of a non-invasive way to form a placenta or to implant. So we've got sort of the embryo, blastocyst, whatever you want to call it, at the top there. All this is the uterus. This layer here is the uterine epithelium. So that's just the single cell layer that lines the uterus. And then deeper into the uterus, we've got kind of stromal tissue and then maternal blood vessels that also have their own lining of endothelium. So in a lot of species, including a lot of marsupial species, the uterus the embryo adheres to this uterine lining, and that's where the placenta will form. That's implantation. But in some species, including us, including rats, the embryo doesn't just adhere to the uterine epithelium, it breaches it. It kind of burrows down through the stroma, and it even breaches the endothelial tissue. So there's direct contact between maternal and embryonic bloodstreams. So this is highly invasive and this is non-invasive, but there's also all kinds of variations in between. And so whether you have highly invasive or non-invasive placentation, in eutherian mammals, we know that implantation can't occur without remodeling of the uterus. So the uterus is kind of a special site that most of the time is quite fortified to prevent things like pathogen invasion. But in order for pregnancy to happen, the uterus has to be receptive to implantation and kind of let down its guard, if you like. So there's only a really brief window that things can actually implant and invade into the uterus, and that's just after fertilization. So normally the uterus, most of the time the uterus is not receptive to implantation, and that's what can cause a lot of infertility, is that the uterus just isn't ready at the time that it needs to be. Because there's this whole lot of morphological and molecular changes that need to happen to the uterus in order for the embryo to be able to implant. So on this side,
we've got just a single cell from that epithelial lining, that layer that lines the uterus, of a non-receptive uterus. So these cells have these long microvilli. This is the top of the cell, this is the bottom, kind of long finger-like projections that we call microvilli. It's also got all these sort of dots around the edges, this lateral membrane and the basal membrane, and these are junctions that are holding this cell to its neighbors, but also to the underlying tissue. So it's pretty well secured in there. With a receptive cell, we've got a few differences. The first is that we lose these projections. So now we've got a nice flat surface that the embryo can adhere to. It's kind of ready for adherence. And we also lose most of these junction points. This is an invasive, this is preparation for an invasive placenta. So loss of these junctions mean that an embryo can attach, and then it can sort of send projections down and under and sort of remove that layer so we can burrow deeper and contact the maternal blood vessels. There's quite a lot of changes that have to happen. And I guess that's all well and good if you're laying down the foundations for a really long pregnancy, like a 9-month one like ours. But what if your placenta is only going to function for a couple of days? So not surprisingly, the short pregnancies of marsupials also mean they have a really short-lived placenta. So this is the fat-tailed dunnet again. This is gestation. So this would be fertilization here. This is embryonic development leading up to birth. And so you can see that implantation and placentation happens pretty late, about 10.5 out of a 13.5 day pregnancy. And that's because for most of this time, the embryo is surrounded by a kind of shell. It's not like a reptilian or an bird egg shell. It's a non-rigid shell, but that shell prevents apposition of embryonic and maternal cells. So it actually has to hatch from this shell in the uterus before it can implant. And that happens here about day 9. So this is an embryo that's squeezing itself out of its shell. It's hatching. So this side is free. This side is still kind of stuck in this non-rigid shell. And it's only after that that the embryo can actually implant and form a placenta. So it's only really this window here, this pink window, when the placenta actually even exists and can function. So that, not surprisingly, again, doesn't provide a lot of nutrition during the pregnancy. So it's only functioning for maybe 3 days. And also because we don't have that blood-to-blood contact, that you can't have that efficient exchange of nutrients in a marsupial species. So instead, nutrients have to be, I guess, secreted by the uterus into the uterine lumen, which is kind of the space surrounding the embryo. So it's sort of bathed in this soup, and it can take up nutrients that way. And instead, most nutrition is provided during lactation. So what we don't know is whether this remodeling process actually happens in a marsupial pregnancy. So since the placenta functions for such a short time and it has quite a different function to most other mammals, do you still need to overhaul the whole uterus in order for it to function? And the short answer is yes, you do need to still remodel the uterus. So what we've got here is, this is the fat-tailed dunnet again. This is looking down on the surface of the uterus. It's a scanning electron micrograph. So each of these kind of irregular shaped blobs is one cell. So we're looking down on the top of it, on the apical surface. So in the non-receptive uterus, we have a lot of cells with long cilia. There are other cells with kind of blobby microvilli. And the surface itself is pretty uneven, and there's kind of lots of debris and stuff in there. So this uterus is not able to accept an embryo. But if you compare that to the surface of the uterus in a receptive uterus, these are really well-ordered cells. They're much more flattened. Their apical microvilli have reduced. So this is now a nice, smooth, flat surface for an embryo to adhere. So because this happens in both eutherian mammals and marsupials, it seems to be a really conserved trait of live-bearing mammals that needs to happen for pregnancy to be successful. But what's not clear is what happens to the junctions in these species, in marsupials, because this is something that needs to occur for highly invasive placentation, and marsupials don't have this. So I was interested in what happened in marsupial basal membranes. So these images are from a rat. I apologize for the quality of them. I stole them from a paper. So the top image represents before implantation, so a non-receptive uterus, and this is a receptive uterus. And this is instead a cross-section through the uterus. So this line here, labeled with a U, is the uterine epithelium, and this is kind of the stromal tissue underneath. There's another layer of epithelium here, sort of both sides of the uterus. And the lumen is just the space in the middle. And each blue dot represents a cell nuclei. And in this case, green is a molecule called talon, which indicates something called a focal adhesion, which are the junctions that hold the epithelium to the underlying tissue. So in the non-receptive rat uterus, we've got this really strong green band, this band of talon localization right at the bottom of the epithelium. And so that's telling us that there's also focal adhesions here and that the epithelium itself is really well attached to the underlying tissue. But at implantation, we lose that layer of talon because we've lost focal adhesions. And this is a species that has highly invasive placentation, so the uterus needs to get less well attached to its surrounding cells so the embryo can burrow in and invade. So that's kind of what we would expect in something like a rat. But what about a marsupial? So this is a similar image again, uterine epithelium here, 2 layers of it. So we've got I hope you can see this at the back, but we've got this green band of talon at the base of the epithelium. So we've got focal adhesions here, we've got attachment between those cell types. But right at implantation, instead of getting a loss of talon, we actually get more talon. So here's that green localization of talon in connecting the uterine epithelium to the underlying tissue. So this seems like there's more focal adhesions here at implantation, which is not something that we would have expected. So it seems like talon's important in both species, but the kind of direction of expression, it seems to be the opposite in a marsupial. It's actually serving to sort of strengthen the connection between the epithelium and the other tissue. So marsupials generally have less invasive placentation or non-invasive placentation compared to other mammals, and potentially that's because the uterus has these defenses against the embryo, which sounds a bit strange, but bear with me. So the epithelium is really the first line of defense against an invading anything, including an embryo. And so the fact that this connection is strengthened just before implantation suggests that the uterus kind of resists invasion to some extent. So the uterus becomes harder to invade in these species at implantation, and potentially this is an example of the uterus restricting or even preventing invasion at all. But why would you want to do that? So blood-to-blood contact is a super efficient way of transferring nutrients from the mum to the embryo. So why would you want to prevent that kind of contact and invasion? The answer is that having that kind of connection between bloodstreams is also really, really risky for a number of reasons. The first of which is that the embryo is genetically, technically foreign. It's genetically half mum and it's half dad. And the mother's immune system can react to that foreign paternal aspect of the genome and can potentially reject the embryo. So in a lot of cases where you have extended pregnancy, like most eutherian mammals do, you also have to suppress the mother's immune system and she becomes immunocompromised. So marsupials kind of get around this by just having such short pregnancies and avoiding that blood-to-blood contact so they can evade detection by the immune system. The second reason why you might prevent invasion and blood-to-blood contact is that where the embryo can contact maternal blood, it can also manipulate mother's physiology, potentially in ways that are harmful to the mother. So it's that paternal aspect again. I feel like I'm always picking on dad, but there's a genetic conflict of interest between mum and the embryo. So in mum's opinion, it's best to put resources into this current pregnancy so it's successful, but just enough so it's successful so that you can retain more resources for future reproduction. But from the father's perspective, he doesn't care about her future reproduction. He cares about this one because this is where his genes are. And so he wants as much resources as possible put into this pregnancy to make sure it is successful. So that's, I guess, a conflict of interest. So where the embryo can actually directly contact mum's bloodstream, it can secrete signaling factors and other substances
that can change mum's physiology in ways that might be destructive to her. And in humans, this can manifest as things like preeclampsia, where the embryo, this sounds anthropomorphic, where the embryo is effectively saying, increase blood flow across the placenta, I need more blood flow, and that can elevate mum's heart rate to dangerous levels. Or things like gestational diabetes, where there's this, this signal from the embryo to increase blood sugar to potentially dangerous levels for mum. So marsupials kind of get around this by, again, not having that blood-to-blood contact, but also pushing this most of the nutrient provisioning to lactation, which can't be manipulated because the embryo is now outside of the mother's body. And lastly, I'm sure a lot of women in the room can attest to the fact that birth is a lot more risky and dangerous when you have to separate two bloodstreams. So again, marsupials get around this by giving birth to something that's incredibly tiny relative to body size and without needing to separate that blood. So all of this to say that marsupial pregnancy is a different way of having a pregnancy that effectively affords more maternal control. And that sounds pretty good to me. So that's the end of that story. So for the last bit of time that I've got left, I just want to take you through one other project that I'm working on right now, which is quite different. We're going to go to the males now. I'm just going to take a second. So this project is at the University of Otago. I've recently got a 3-year grant from the New Zealand Royal Society to work on seasonal biology of the marsupial prostate. So if you are a mammal that breeds seasonally, your reproductive organs are usually only active for the part of the year when breeding can occur. That's because it's energetically costly to maintain things like a testis or a prostate in an active state all year round when your chances of breeding are 0 for the majority of the year. So it doesn't make evolutionary sense to keep them active the whole time. And so what that usually means is that during the breeding season, however long that is, your reproductive organs are generally a lot larger and they're more active. They're secretory, if that's what they do. And then outside of the breeding season, they really reduce in size and they become inactive. That's kind of a cost-saving measure. So this is an example of testis size in the roe deer. So this green box here is showing where the rutting season is. It's kind of mid-July to mid-August. So we've got change in, I guess, relative size of the testis here, and then this is testis volume. So you can see that the testis is at its maximal size during the rutting season and just a little bit before and after. But over the winter time, the testis is a lot smaller and it sort of has to actually increase and activate in order to get to those reproductive levels before it regresses again. So this is a pretty common pattern that happens in reproductive organs in a lot of seasonally reproducing mammals. And I guess the other side to this, as well as the testis function, is also prostate function. And that's something that hasn't been looked at as much. And I mentioned kind of at the top of the talk that in marsupials, prostates are actually really important to male fitness because they're the only organ that marsupials have that can produce seminal fluid because they don't have seminal vesicles. So it's that sort of carrot-shaped organ there again. And marsupial prostates in the species that breed seasonally also undergo a pretty dramatic shift in both size and activity over the year. And that's particularly true for the brush-tailed possum. So this is prostate weight in the brush-tailed possum. So possums breed in the southern hemisphere autumn, so kind of April to June-ish. So you can see that in sort of November to January over the summer, prostate weight's at its lowest. It's kind of less than 10 grams. That probably doesn't mean much to you, but going into the breeding season, it's increased more than 5-fold, which is a big change for an organ. And so this is an example of an inactive summer prostate. It's just a normal petri dish. Sorry, it's a bit gross. And this is an active prostate. So this is February, and this one is April. So that change happens in less than 2 months, and it's just a massive increase in size. These things are huge. Possums are only about this big, so they represent a huge amount of their body weight during the active breeding season. So I guess the two things that are important to know is that this change not only is quite a
substantial change, but it happens really fast. And it's a combination of both cell proliferation, so there are more cells in this organ than this one, but also cell enlargement. So the cells in this organ are bigger than the cells in this organ here. And if we have a look at the glands within this, the prostate it produces seminal fluid, it's a secretory glandular organ. In the inactive prostate, these glands, which are kind of the white circles, are pretty small and clustered together. There's not much stuff inside them. It's kind of a different cross section here. But in the active prostate, these glands are much bigger and they're full of that liquid that they're about to secrete. So that kind of amount of change and the speed of which these organs are able to change is pretty unusual in adult tissues. We don't hear of things like liver or kidney undergoing that kind of change. And if we do, it's usually in the context of disease or things like cancer. So what we've got here is an example of rapid growth that is controlled rather than uncontrolled, like you'd have in tumorigenesis. So this is a way of investigating some of those mechanisms of controlled growth in a species that does this as part of a healthy adult tissue. And what was amazing to me is that we really have no idea how these prostates are able to change. So what the genetic mechanisms are that underpin these dramatic shifts in morphology. And we don't know this not only for marsupials, but for pretty much any mammal. There's a little bit of preliminary transcriptomics data for one species of ground squirrel. Can't remember what the species is, but it was just a preliminary pilot study. And besides that, we don't actually know what's going on in these organs at the genetic level. And what I'm particularly interested in are the genes that are involved in going from an active maximum size down to your minimum size. So involved in sort of controlling of growth or regression. That's because there's quite an interesting link between brush-tailed possums and a very common human disease called benign prostatic hyperplasia. So this is not prostate cancer, but this is growth of the prostate. So humans have, this is the bladder, this is the prostate. Humans have this kind of weird prostate anatomy where the prostate completely encircles the urethra. And that's kind of a poor design because as the prostate expands, it compresses the urethra and it results in all kinds of urinary symptoms. And it's really common with age. So unlucky for possums, they have this same unusual anatomy, which is really quite rare in mammals. There's only 3 or 4 species that have this prostate that circles the urethra. And so the way that this prostate enlarges seasonally during normal reproduction mimics the kinds of changes we see in a disease context in a human. So the growth happens in the same place. The anatomy is representative of a human. So if we can figure out how this growth is controlled and reversed, that would have potentially clinical relevance to human prostate conditions. That's what I told the funders anyway. So I'm interested in understanding what gene expression patterns underpin these changes in prostate morphology and kind of what categories do they fall into. We don't really even know if it's things like proliferation or apoptosis that are causing the changes, or maybe something else is happening. And is there much overlap in gene expression in the possum prostate to what we already know about human prostates? So we know that there's some pretty good morphological evidence that possums are a good model or could be a good model, but we don't know if we can find that same kind of similarity at the genetic level. Let's see if this will work. You didn't know I put a video of you in this talk. So I just want to show you where we get our possums from. So at Otago, we're super lucky now to have a colony of brush-tailed possums that was set up by and basically completely run by Dr. Richardson. So I've just got this kind of cute video. There might be sound. I don't really care if there isn't. So you'll see, embarrassingly, that he's wearing a Montana Tech sweater. There's 3 in a box. So cute. These are all females, so we didn't collect any prostates from them. But we've got this really great setup to do marsupial research now with this colony. If there was Sanyud here, they were grumbling and hissing and not into taking apple that day. I think they're really lovely. I'm supposed to not like them because they're a pest in New Zealand, but I think they're adorable. How could you not like those faces? So I don't have a huge amount of data to show you. I'm just at the beginning of this project, but I was really lucky to have a fantastic honours student this past year, Josh Plowman, who did a pilot study to kind of get a real basic idea of the gene expression patterns that we want to see. So what he did was he made RNA-seq libraries from a handful of active and inactive prostates, so kind of the top of the peak and the trough, so from April and December. And what RNA-seq or gene expression or transcriptomic studies do is they give you a snapshot of all the genes that are expressed in that tissue at that time point. So then you can compare, say, active versus inactive gene expression profiles and see what's similar between the two. That's probably stuff related to just being a prostate, but more importantly, what's different between the two. And that can show you what's changed between those two time points. And given his time constraints, we were only able to do pretty low coverage sequencing. So I think we had 20 million reads in total, which really isn't very much for this kind of study. We'd want a lot more than that. But even at that level, we were still able to get some really cool results. So this is a scatter plot. Oh gosh, you won't see this at the back, but there's lots of gray dots in the middle. So every dot represents a gene that was expressed. And the ones along the axis are ones that were not significantly different between active and inactive prostates, but the ones in blue are different. And so the blue dots that I've circled in red are those that were significantly higher. They were expressed, what do I want to say? They were overexpressed in the inactive prostate relative to the active one. So they were functioning in the inactive prostate. And these blue dots circled in green showed higher expression in the active prostate relative to the inactive one. So we actually found 56 differentially expressed genes, which is a heap for low coverage data. So that's giving us this really good idea that there are a lot of genetic differences and we're only just scratching the surface of what those are. So 26 of those were upregulated in the active prostate and 30 of them were upregulated in the inactive prostate. So that's showing us that there's kind of different functions that are happening in these two, the same organ, but at different time points. And Josh was able to find out that about half the genes we identified had been previously found to have roles in either benign prostatic hyperplasia in humans or prostate cancer, including 11 that were related to ribosomal function, which is really important for cell growth and activity and proliferation. And this tumor-related gene called TCTP, which is a regulator of cell growth and proliferation, and it keeps coming up in prostate-related diseases. So this is some pretty good data as a preliminary set, but given that I've just kind of begun this project, there's a lot of next steps. And the first is to include a lot more time points. So we just looked at the peak and the trough, but what I'm really interested in are these genes that are changing as the prostate is activating and becoming inactive. So those are going to be where the kind of interesting biological phenomena are happening. I've just got some data back that I haven't looked at. That's about 10 times the coverage of what I've just shown you. So that will be a little bit deeper, but we'll be able to flesh it out more when we've got a lot more samples across time points. And this is part of a bigger study looking at seasonal reproduction more broadly. So this is obviously a marsupial, and it's hard to compare that to something like a deer. It's sort of apples to oranges. So the plan is to have two pairwise comparisons. The possum, which is a seasonal breeder, with the opossum, which does not breed seasonally, and that will help tease apart which genes are kind of marsupial related and which are specific to seasonality. And then the other comparison is between a deer, which reproduces seasonally, and something like a rat, which doesn't. So by comparing, this will be a lot of data, by doing all of these different comparisons, I'll be able to tease out what genes are specific to different clades, and within those clades, which genes are involved in seasonal breeding. And once I've identified some kind of interesting key genes related to growth or aggression, whatever that looks like, I'll test these functionally in cultured cells by either knocking out or overexpressing those genes in cells from active and inactive prostates and seeing how that affects their growth. So I hope that I have convinced you that marsupials are pretty awesome research models, and that where you've got really unique biology and a cool evolutionary placement, there's actually heaps of potential for discovery.
And thank you so much for listening. And with that, I'll take any questions. It's also cool if there's no questions. Oh, yes.
So things that are cycling, whether they're annual cycles of testicular size or whatever, they undoubtedly have a hormonal basis to that.
Yes.
Which is turning on and off genes.
Yep.
And so I think, does anyone have, there must be some data available for the hormonal cycling.
Yeah.
Marsupials and mammals that are showing this, regression, recrudescence of testicular size, for instance. There must be some data.
Yeah, you're right. So prostate is the one that I'm interested in, and it's an androgen sensitive organ. And so definitely things like testosterone are playing a role in mediating those changes, not just in the possum, but in a whole range of species. And so we know that things like androgen receptor change in expression and localization during the breeding season. I guess what I could have probably said better is that we've got a little bit of data from candidate gene studies looking at specific genes like how does androgen receptor change. But what we haven't done before is profile all of the genes involved in these processes to look not only for the things that we expect to change, but other stuff that could be contributing to that. So another example is marsupials don't have oxytocin, they have a different version called mesotocin, and mesotocin receptor also change expression over the breeding season. But those are kind of the only, that plus like one other gene are the only ones that we know of that change because they're the only ones we've looked at. So what I'd like to do is look at all the genes and then kind of from there pick out ones that are significantly correlated with this change and see what they might be doing. Yeah.
Do we know what controls that change? Keeps the clock on time?
Yeah, so it's a combination of both. So I'm just going to talk about the possum because that's what I know the best. It's a combination of day length and female receptivity. So female organs also shut down, which is something else I'd really like to study. So possums had this anestrus period where they're not cycling over the summer. And then as the days start to get shorter, the estrus cycle starts to kick back in again. And that's a trigger for the males to undergo these changes. So we kind of know, I guess, those parameters, this level of change. We know that things like day length will change it. We know that things like exposure to females can change things, but we don't know kind of at the molecular level how that change is actually implemented in the organ.
So if you had some captivity, could you control the amount of light they're exposed to?
Absolutely, you can do that, and you can shift breeding that way. Yeah.
It looks like your cycle for reproduction is the opposite of the northern hemisphere because they're moving winter.
Yeah.
It looks like our summer months, but that's your winter months.
Yeah.
And is that because of rainfall or why are they?
Yeah.
Nothing bleeds pretty much in the winter in North America, but.
Yeah. So in Australia, where possums are from, but in the southern hemisphere, summer is usually the most extreme season. Summer is the season of drought. It's the most erratic with rainfall, as you say. So summer is the season that you really prepare for, and it's when most things shut down. So our winters are pretty mild and temperate. So it's sort of like 40 or 50 degrees in the winter. So usually there's not much of a restriction there. You don't have to protect something from incredibly freezing cold, but you do have to protect something from massive heat. So it's better to wait until after that before you start to kick into reproduction and breed over the winter months. Yeah, Michelle.
In that first study, for that species, how many pregnancies would a female typically have? And then with those pregnancies being so short, and I'm not quite sure I'm getting the anatomy right, but are there other things going on in the uterus outside of the period of pregnancy?
So do you mean, does the uterus change at all during the estrus cycle? Is that what you mean?
I guess, is there any other functionality? I wasn't sure how it related to.
Yeah, so normally that species would have just one pregnancy a year, maybe two, if something happened to the first or conditions were good enough that they could breed a bit later on. So even though they have such short pregnancies, they typically still only have 1 or so a year. And that's because the suckling stimulus of the young in the pouch usually prevents further ovulation. But if you were to remove that, they can get pregnant again pretty quickly. But I think I kind of understand your second question, which is that during the estrous cycle, there's some amount of change. So the uterus kind of half-heartedly prepares for a pregnancy. But if there isn't that signal there from the embryo itself to say, I'm here, this is really happening, then that will just sort of regress and go back to normal. So there's this sort of like a little bit of change that happens where you sort of get the beginnings of that change, because it does take quite a while for the uterus to remodel. So you want to have begun that before pregnancy is confirmed. But if pregnancy is not confirmed, then it's easily reversed. Is that kind of what you were getting at? Yeah. Yes.
I get this aspect about extra embryonic membranes. We have the amnion, corona, elicus, and yolk sac. Do those same four occur in marsupials, and to what extent are they? Is that abbreviated or something? Because they're very important in making the descent.
They are.
Experience, but maybe they're not important at all.
No, they're still important. So those membranes are pretty conserved, even across even into reptiles and things. But which one is sort of favored differs between different vertebrate groups. So marsupials have what's called a choriovitelline placenta. So it's mostly the chorion that's forming rather than the amnion. So it's the chorion that does most of the work, and it's the bit that actually implants. So that's the kind of difference there. They still have all those membranes, but it's that side of it that actually is the dominant one in marsupials compared to other mammals.
So they're still within an amniotic fluid.
They are, yes.
They
Yep.
just don't go very far.
No, they're sort of prevented from having that invade or get too elaborate, if you like.
I want to ask a question on the molecular stuff. So there's got to be a lot of research out there on gene expression patterns in tumor versus non-tumor prostate tissue in humans, I'm assuming.
Yeah.
They've done microarray analysis or RNA-seq. I know your data are preliminary, but have you seen any gene overlap with genes identified in those studies with what you're seeing here?
Just a little bit. So
Okay.
so sometimes when you look up a gene or what does this do, for quite a few of the ones that we found out of those 56, the only thing that was known about them was that they had come up in something like a tumor cancer microarray. So we don't know what else they do, but we know that they were implicated in that sort of study. So I mentioned one called TCTP. I can't remember how that goes now. So that's a tumor-related gene that's kind of specifically upregulated in prostate cancer rather than other types of cancer. So it's interesting to see that here, because it's conserved apparently between marsupials and eutherian mammals, but this isn't cancer, this isn't a tumor. So it must do other things than just be part of prostate cancer. But I guess it's been co-opted in cancer to do, it was a proliferative gene. But I guess in some ways it's been harnessed for proliferation here, presumably, but in a way that doesn't automatically lead to cancer. So we'll get a lot more of that kind of data, hopefully, as it gets deeper and we've got more time points. But those are the kinds of things that we're interested in kind of mining the data for. Yeah.
All right. So today in Dr. Richardson's class, he mentioned that
Here we go.
kangaroos can sometimes have two joeys.
Yes.
So how is that really possible then, since they mainly just have one every year?
That's a good question. So some marsupials have quite a lot of offspring at once. The Tassie devil has something like 30, but they only have 4 teeth, so there's massive competition there. But the wallaby, the Tama wallaby in particular, is an example of a species that just has 1 offspring in 1 pregnancy per year. But he didn't lie. So they can actually have 3 at a time at different stages. So there's 1 in the pouch that they've just given birth to. They've got 1, they call it a young at foot, so the one they've just kicked out of the pouch to make space for this new one, because it's about 10 months old by that point. And crazily, The Tamar wallaby is a really weird species that gives birth. They all give birth on the same day because it's photoperiod driven, and they mate on the same day. And then that embryo develops to the unilamina blastocyst stage, so only kind of a few days in, and then it goes into embryonic diapause, and it's held there in kind of suspended animation by the suckling stimulus of the young in the pouch. So they have kind of 3 on the go at once. They've got one that's basically weaned, one they've just had, and one that's kind of in the oven ready to go. And these animals live in pretty variable, unpredictable, dry environments. So they can, if things got really bad and there wasn't enough food, they could tell their weaned one to, go and fend for yourself. They might sacrifice the one in the pouch, and there would be 2 to 3 weeks before that would kickstart development of the one in suspended animation. And so in 2 to 3 weeks, they would have a new baby, and it's a spare, I suppose, if you like. Are there any more questions?
I'm gonna call this before I get thrown under the bus.
Call it.
Thanks for coming, everyone. Thanks, Mel.